I'm going to talk about our efforts to reconstruct the placental immune response to bacterial infection. So we use, as Siko mentioned, multimodal imaging to interrogate bacterial infection that affects children mostly. So we are using these techniques in animal models of bacterial infection, prenatal infection, and cystic fibrosis, other infections that affect children. We use a multimodal approach using these various techniques, PET, SPECT, MRI, intravital microscopy, just about everything we can get our hands on. Here's a computed tomography, MRI, and PET SPECT. This is CT PET. We use a bunch of different techniques. Our main go-to is in vivo bioluminescence imaging. So this involves the Lux operon that we label the bacteria with. So this encodes the luciferase, which produces the light, and also the substrate for the luciferase is made by the same operon. So these bacteria spontaneously glow. We detect them in a live animal with this instrument here, the IVIS instrument, in vivo imaging system. And here's an image of Pseudomonas in the throat of an animal. So the main prenatal organism that we study in regard to placental function is Listeria monocytogenes. This is a gram-positive bacterium. It's an intracellular bacterium, so it grows in the cytoplasm. It breaks out of the of the phagocytic vacuole and swims in the cytoplasm of the cell. It can spread from cell to cell using actin-based motility. It elicits strong immune responses, innate and acquired immunity, and it's one of the most well-characterized immune models of all of bacteria. And it's currently being used in my lab and also in other labs and in clinical trials as an anti-cancer treatment because it elicits strong immune responses in the tumor microenvironment. So we're working on, that's one of our projects is Listeria as an anti-cancer treatment. Prenatal infection model using bioluminescence imaging. Listeria infects the placenta. This is the immunology or the immune response we were talking about here. The placenta lights up very brightly with infected Listeria, but we get a different range of different infections depending on the dose and timing that we use. We can get a very mild infection or very strong ones, such as seen here. We then interrogate these animals with MRI, with ultrasound, and with other techniques in order to see the effects of this placental infection by Listeria. Now, if you look at the placenta, the structure of it, it's truly an amazing organ. It's unique in all of biology, really. And so it varies a bit difference between human and mouse. And you see these structures here. As we all know, the trophoblast plays a key role here in contact with the decidua, which is maternal in origin. And so the immune response of the placenta is a fascinating subject. It's very understudied. It's not understood well how these two tissues, which are derived from genetically different sources, can interact and that's what makes trophoblast such a special cell. And so there are these different structures here. In terms of Listeria, in the human placenta, there are these extravillous trophoblasts that are in mom's decidua. And so that is the access point for Listeria in the human system. Listeria is an intracellular pathogen, so it comes in other cells and invades these tissues in this way. Let's look at that a little bit more closely. So you see these extravillous trophoblasts. Listeria comes in an affected monocyte or macrophage, mostly monocytes in the blood. It can then spread to these EVTs, these extravillous trophoblasts, and that gives it access to the syncytium. And so the syncytium itself, when it forms, it will block Listeria infection. Listeria can't get through that in syncytium. And so the only way you can get access is through this sort of indirect method of infecting the trophoblasts in that way. If the syncytium is damaged, then Listeria can cross there. So what we see in our bioluminescence imaging models of this is placental infection by Listeria. Here's a big old pregnant mouse showing the placental infection here. We took this mouse and excised the left uterine or the mouse's right uterine horn. So that's this one. You see no signal. And then from this other side, you see all these infected placentas here. And this is the system we use to study the immune response of the placenta. And so here's the excised, one of the excised fetuses, not from this animal, from another, but you can see this infection of chorion. So this is fetal derived. The decidua is not shown in that yet. Anyway, in some of the animals, you can get this crossing, as I mentioned, into the fetus through the trophoblast syncytial layer. Here's an animal here that with a restricted to the placenta, and then here's one where it has spread into the animal through this method. It's dependent on the Listeria protein internalin P, which our lab discovered has a great many variants and is a very interesting protein. It's necessary for this crossing event to occur efficiently. So we are looking at the placental response to infection. This was done by Kayla Connor in our lab. So we can excise these uterine horns and extract RNA from this placenta or that placenta. We can look at the different placentas and compare them from within the same animal. So this is a very nice control. That controls for any systemic effects that they might have, because those should be seen in the uninfected placenta. And then we look for localized effects in the infected ones, and we can compare them. And so one of the things that Kayla found right away was altered arachidonic acid metabolism. So these are various prostaglandins and lipoxins that are elevated in the placenta. This is a part of the eicosanoid pathway and arachidonic acid metabolism. So you see phospholipase A2 here is upregulated, and COX-2 and other really well-known members of this pathway. And we see these upregulated regulatory molecules, the prostaglandins, and those are involved in, for example, preterm birth, which we see in these animals. And so we get a bunch of different of these molecules upregulated. So Kayla coined this the icosanomes. So I thought that was a cute name that she came up with. Anyway, so these have multiple important functions in the body, and so to find this was upregulated was not really a surprise, but it was very interesting. Now, so if you look at infected and uninfected, so infected ones on the left and uninfected on the right, you see the degree of infection is associated with the degree of these upregulation of these molecules, the leukotrienes and the prostaglandins. So then Kayla also looked at the transcriptional response to the placental infection. We see these upregulated genes and some downregulated genes. I'm going to come back to these downregulated ones. They're very interesting, but let's talk about the upregulated ones because they relate, I believe, to the previous talk, and I'm so glad that that person talked about that. Anyway, so here is the infected and uninfected. So these are upregulated genes here, and these are the downregulated ones. When we look at almost all of these are upregulated by interferon gamma. So those are almost all of them are interferon gamma responsive genes. And if you look at some of the functions of these, they're involved with the immune response, as you would expect. And so these are all upregulated by interferon gamma. A couple of these have interesting activities that we're looking at in the placenta. One of the things is there's not time enough in the course of infection that we've shown that we use here. We infect on day 14 of the 20-day gestation of mice, and then we analyze them on day 18 or let them give birth. So that's not enough time for cytotoxic T cells, antigen-specific cytotoxic T cells to respond. So we believe that this is all NK cell mediated, and NK cells are crucial in placental function. They're crucial for the development of the placenta, and they're crucial for the immune response of the placenta. So we believe this is NK mediated, all of these genes, and some of them are interesting. For instance, this one encoded by the GM12250 gene, this IRGB10, is responsible for sensing bacterial DNA and then in activating the NLRP inflammasome. And so could be very important in the placenta. Another really interesting one that's highly upregulated is IDO. So this is indoleamine dioxygenase. This metabolizes tryptophan. When I first saw this, I said, well, so what? What does this do? But it turns out the product of this pathway, one of the products, kynurenine, is super important for the immune response and also for development. And it's just a very important metabolite for many, many different purposes. So this is a very significant pathway to be upregulated. And also tryptophan is metabolized to serotonin, which produces serotonin is derived from tryptophan, and placenta supplies the fetal brain with serotonin. So this could be very important for some other things that I'm going to go into. Here's the expression of IDO. This was done by these people. It's found in the villi of the placenta. So a very interesting upregulation. And when we look at, we can do image-guided mass spectrometry. So we take some of these placentas and look and see what's in them. And sure enough, we find elevated kynurenic acid. So this is related to kynurenine, which is one of the metabolites in the IDO1 pathway. So we're very interested in that. And one of the reasons we're interested is we see altered brain development in these animals. So here's within one uterine horn here, or one set of fetuses in the same animal, and Justin Lee did this work. You can look at the fetal brains here of a fetus that is from an infected placenta and one from an uninfected. And you see that the layer from the uninfected one has these nice, or the brain layers are nicely organized, and this is normal brain structure, but these are disrupted. You can see the layers are not quite normal here. They haven't formed these normal layers. So we see abnormal brain layering. Also, you can look at the gene expression patterns of these brains from an infected placenta and one brain associated with an uninfected, and you see sex-specific alteration of gene expression levels. And this was what Justin found. I'm not going to go over this too much. But the sexes of the fetal brains do show differences. For instance, here's HIF1 transcription factor upregulated in males, but not in females. So that's an interesting thing that we want to look at, the sex specificity of these interactions and the immune response in the placenta. Does this occur in the placenta, or is it in the fetal brain, or is it a combination of both? And we don't understand that yet. So then we can let these mice give birth and then analyze their behavior. So here we've shown here this three-chamber setup that Justin used. You put a mouse in the middle here, and then you put a roll of tape over here on this side, and then you put a mouse on that side, and then you measure the amount of time that they spend in each of these chambers over time. And most mice will prefer to spend time with a mouse in the chamber rather than an inanimate object, but mice with neurodevelopmental disorders spend more time with an inanimate object, and that's called social interaction. And so we can measure that, and we measure some other behavioral effects of these changes, and we see that they're sex specific. So the alteration is sex specific. We see social interaction is affected by listeria exposure in males, but not in female. And we also see the grooming is highly elevated, and this is a sign of repetitive motion in males, but not in female. On the other hand, we can remove all of these items from here and just watch the mice explore this space, and that's called open field exploration. We see that the control females exhibit normal behavior in this way. The affected females exposed to listeria hug the wall of the chamber, and that's a sign of elevated anxiety. And so those are greatly affected in females, but not significantly affected in males. So we see sex specificity in this. Now I'll go back to the down-regulated genes in the placenta. Now, some of these we don't know what they do. Of course, we have some immune suppression that's down-regulated because we're getting elevated immune response, so that's expected. But here we see some of these that are related to neural development. For instance, these genes here, which are downregulated, are all related to neural development, and mutations in these genes cause neurological effects in humans. And so we're very interested in that because this is in the placenta. It's not in the fetal brain. This is the placental response. So we're very interested in how these would affect fetal brain development. And some of these genes are related to autism. I'm not going to go into that. This is not Tylenol. This is an infection. But anyway, we see these genes are related to genes in the autism spectrum, and the sex specificity resembles human autism. By the way, several prenatal infections are associated with human autism. So here's what I wanted to get at. We've got these cells from accelerated bio. These are the HTSCs that they talked about in the last talk, and iPSCs from these HTSCs. Now, what we want to do is differentiate these into NK cells. So I would love to talk to Dr. Papasaba about this because we believe that the NK cells are making the gamma interferon that is inducing all these genes. And we would also like to make the monocytes that get infected with listeria, we could make those from the same iPSCs. Now we can make syncytia induce the formation of those and maybe make extravillous trophoblasts or cytotrophoblasts, giant cells, all the other trophoblast type cells, and then make placental organoids from these and infect them with listeria. And that's what we would like to do. Then analyze the NK activation status, the TSC responses, the sex specificity. Now, if we could possibly, I don't know about this, but to get male and female HTSCs and compare them, a set of male ones and a set of female ones to see if this sex specificity lies in the placenta, in the immune response of the placenta, because this is a known thing. So people are looking at that kind of thing. Now, one of the reasons we want to do this process is that our previous grant to the NIH was all animal work. And so we need to have an in vitro component of that. And so this will work perfectly for us for that grant because NIH is now saying they won't fund work that's all animal. So this is a nice in vitro model that we would love to make. And so we're talking with STEM Matters to get the iPSCs that were derived from that cell type. And so we got the MTA on the way. We really want to try this. I think it would be very interesting. We can not only do we make maybe these organoids, but we could use transwell assays or, co-culture or all kinds of different things. I didn't talk about our work with EVs because I talked about that last time in the last session, but John Koletka in my lab has made EVs from TSCs, and they exhibit altered immune activation and properties due to damage-associated molecular pattern. Very, very interesting result that he got, in my opinion. And so far, we've done this. Here are the cells. I mean, you saw them in the last talk, and here we induced with a CHUR compound these syncytia to form, and you can see them. They have multiple nuclei inside there. So these are syncytia, and we're going to test those with human chorionic gonadotropin that's elevated in syncytia. So we're in the process of doing that. And so I'm very excited about differentiating these cells and co-culturing them and making NK cells from them. And so that's what we're going to do. And so I would like to thank my grad students and especially Pramika, who's an undergrad helping me grow these stem cells, grow the stem cells that were provided by Accelerated Bio. And here's some of the other people that were working with us. So thanks very much.